Okay. Thank you all for staying on for this second session of the advanced courses. My name is Ku. A little bit about this course is that we just heard about the glycan binding proteins, and obviously the glycan binding proteins binds the glycan. And for lots of you, everybody is very familiar with N-glycosylation, hybrid type, high mannose, complex type. But I think most of you should also know a lot about O-glycosylation. And it's not just the mucin type O-glycosylation, O-GalNAc, which obviously Henry is going to talk a lot, but also to me a very fascinating field of O-glycosylation, and there are more variation on it, and particularly the O-glucose, O-fucose, and etc., which Bob is going to talk about. So I have the privilege here to introduce the two speakers. I'll introduce more about Bob after the coffee break. About Henry, he's a guru of this field, and I think for most of you, younger generation, you obviously know that he founded the famous Copenhagen Center for Glycomics. But before that, he did his work in Denmark as a dentist. Is it? Yeah. Okay. And then, but importantly, he went to Seattle and he was with Itaro Hakumori, if you know who he is. And if you don't, you should know him. Okay. Hakumori is the grand guru of our field in glycobiology. So he spent some time in Seattle there, and including being the head of the Department of Immunogenetics in the Biomembrane Institute in Seattle. Then he came back to Denmark, and very soon he founded this Copenhagen Center for Glycomics, which is the main center of excellence for glycobiology. And the other center of excellence in US, obviously, is the CCRC, where Bob is. Okay, so we have two people from these two different centers talking about a very important field that not only we know so much about it, but we also want to manipulate it. And that's where the engineering and CRISPR and everything comes in. So without further ado, I'll pass on to Henry.
Thank you. Thank you, Ku. Thank you very much for organizing this. I think it's a fantastic idea to have an advanced course in front of a very big scientific meeting like this, where students can come and hear more details. So Bob Haltivang and I will share, and we'll kind of float the introduction in, and so we will see us dance in and out during the talk. And I will start slowly here. So I'm from Copenhagen. It looks a little like this, and we are up here, our center. Our center is it may be famous, I'm not sure about that, but it's much, much smaller than CCRC in Athens, Georgia. But we are up here on the 10th floor with a fantastic view over Copenhagen. Although this is the only day they could take the picture where it's partly sunny. It never is sunny like here, and never this hot. Oh, okay, yes. So what we will be talking about is now not binding to glycans, but the glycans. And the glycans obviously are positioned in the membrane on lipids or protein. And what you see here is a different sort of presentation than Matthew did before, which is color-coded into what we call the different glycosylation pathways. So we have a number of different glycoconjugates, and as Ku started out by mentioning, most of you are very familiar with N-glycosylation, which you've already heard a lot about today, also glycolipids, and of course the mucin type or GalNAc type O-glycans. But there are many different types of glycosylation, and we would like to talk about some of the O-glycosylation pathways today. Specifically, we would like to talk about the mucin type, and then the notch, and then this expanding family of O-mannose. And without much further ado, we will do our little dancing, and Bob will continue on the next. I will talk about this and the O-man, and Bob will talk just briefly introduce the notch O glycosylation. I am pressing this.
I will talk about cysteine-rich modules that EGF repeats and thrombospondin type 1 repeats. So these are, again, as their name implies, they've got a whole bunch of cysteines in them, and these two domains have 6 cysteines. I'll explain them more in my talk. But there are actually several dozen cysteine-rich domains in biology, but these are unique because they have unique O-link glycosylation modifications. There are a number of proteins, as I'll get into, that have these modifications on them, but there are several proteins that are the major targets, at least biologically speaking. These are EGF repeats. The Notch 1 extracellular domain has, each one of these is an EGF repeat, the ovals. There's 36 of the EGF repeats, and almost, as you can see, almost all of them are modified with either an O-glucose glycan or an O-fucose glycan. And this is for several of the enzymes, POFOT1, POGLIT1, and EOGT, which I'll be talking about in more detail later. And then there's a homolog of POGLIT1, 2 homologs of POGLIT1 called POGLIT 2 and 3, and they actually add an O-glucose to EGF repeat, but at a different site on the EGF repeat. There's only 1 or 2 POGLIT 2-3 sites in the notches, but in the fibrillins, there are many of them. Fibrillins are extracellular matrix proteins that have about 47 of the tandem EGF repeats, and the ones with the blue circles on top are where we've site mapped those. One of the things that's important that I want to point out is that the biologically important proteins with these modifications tend to have a lot of them, a lot of the EGF repeats with the sugars. This is just an analysis of the phylogeny of the enzymes that are unique to modifying EGF repeats and their substrates, so the notches and the fibrillins. We're looking for homologs of the human genes or the human substrates. And you can see that all vertebrates and insects and tunicates and worms have homologs, and so they're unique to the metazoans, the multicellular animals, but fungi, plants, and protists and all bacteria do not have them, and so these are unique just to multicellular animals or metazoans. For the thrombospondin type 1 repeats, TSRs, again, there are, about 50 proteins that are predicted to be modified with them. ADAMTS9 is one of the most biologically important ones, as you'll see when I start talking. It has 15 of these TSRs, and 12 of them are stoichiometrically modified with this fucose-glucose disaccharide. Multiple TSRs are the proteins that have most TSRs are the most biologically important substrates for the enzymes. And I've done the same thing here. The POFOT2 and beta-3 glucosyltransferase are present in vertebrates, insects, tunicates, and worms, but again, not in plants, fungi, or protists, or bacteria. So this just kind of gives you a broad overview of where these, in biology, where these substrates and where the enzymes that modify them are. And so I'll turn it back over to Henrik.
Thank you. So when we look at all what we call the glycome, so I will, for presentation's sake, limit to the human glycome, but in reality it's fairly similar throughout all mammals. When we look at this presentation of all the different types of glycoconjugates, we obviously see all the different glycan structures, but we don't see the full perspective. So the glycome, in terms of all the glycan structures we can build on each of these different glycoconjugates, lipids, proteins, and so forth, is quite big. I won't say it's vast, although I wrote that, but it is very big. And with many years in glycobiology, I've given many talks where we have used structure-centric presentations. So we just go through all the different glycan structures. And today, I will invert that to a genetic sort of biosynthetic point of view instead and tell you what regulates it. Because while the glycome is really very large, many different structural components, many of the structures that we know are really parts of the same scaffold. So in other words, in terms of how they're biosynthesized, it is much simpler. And this is where I would like to argue that the glycosyltransferases that shape the glycome is a much more limited area to look at. And with viewing the glycome in terms of the GT or the glycogenome, if you want, if you allow me to use that word, prevents some obvious opportunities. Number one, you don't have to be a carbohydrate chemist or glycobiologist to understand what is going to happen. You can infer to a biosynthetic scheme, and then you can work with genes instead, which means, in my opinion, that we can expand the interest of our field way beyond glycobiology as a field. Now, with this view, obviously, you have to look at the biosynthesis. So where are the glycans biosynthesized? Now, I'm sure you're aware of the O-glucanac modification that occurs in the cytosol, and we're not going to talk about that. But otherwise, all other types of glycosylation occurs in the ER and Golgi apparatus of the secretory pathway. What is interesting to think about is when they are initiated. First time when a glycan is added to a protein. I'm sure most of you are aware of how N-linked glycosylation is added with an ensemble of a precursor oligosaccharide and then trimmed down in ER and then further built up. Those are steps we can call initiation when you put the sugar on, and we can call it co-extension, elongation, branching, and capping as the glycans are trimmed, matured, and finally capped, or we can call it processing towards secretion to the membrane. Now, most types of glycosylation are initiated in the ER. That means they occur simultaneously or perhaps before or immediately after or in association with protein folding, which obviously gives a lot of opportunities. It also means that the glycosylation can affect transport from ER to Golgi. O-GalNAc or mucin-type glycosylation is initiated, at least in normal cells, in cis, Golgi, and throughout the Golgi stacks. It's not only in cis, also medial trans, and perhaps even trans-Golgi network, as is O-xylose, which is, in this case, the glycosaminoglycan addition. Now, this occurs then after folding. And that means your substrate sites may be limited by the structure of protein, because in reality, you will have to have exposed the acceptor sites on the proteins. So if we look at it that way, and we think of the biosynthetic pathway, now I know it's dawning, but then we just turned the glycosylation machinery into all the known enzymes, or, of course, glycosyltransferase genes that make up the glycome. And in reality, we end up with something like 175 genes that make up this enormous diversity. Now, I realize 175 genes, and if I ask you to remember where all these are, it will be a little of a problem. But 175 is much, much less than the structural variability you have to deal with as a glycome. Now, in this, we have assigned the genes and their main pathway. So this is by no means resembling all the structural diversity you can in glycans, but it assigns an enzyme to one linkage. When I was trained as a glycobiologist, I won't say how long, but it was in the last millennium. It was a daunting thought to think that there was an enzyme for each glycosidic linkage that we knew. In other words, that you would have enzymes that would specifically do one linkage. And as we came through the genome area up to year 2000, it appeared that we have, for many linkages, multiple enzymes, so isoenzymes. So there's many more enzymes than predicted originally. But we can basically build 175 genes into this system. Now, the reason this is interesting is that we can now look at the glycogenome, if you want, or the whole glycosylation machinery at a global level. And the first thing to do is look at how we arranged. Here you can see it says pathway specific. How we can arrange about 2/3 of the glycosyltransferase genes in the genome, so more than 100, into unique pathways. So all the enzymes, for instance, here in GalNAc type O glycosylation, they only work in this pathway, and so forth. So that means by knowing these enzymes and expression or no expression in a cell, you can turn on or off a particular glycosylation phenotype. There are few glycosyltransferase in this pathway specific that have isoenzymes. Most are single genes. You can see, for instance, we'll talk about the metroglycan biosynthesis, the N-glycan biosynthesis, and to a large extent also in the O-fucose. There's only one gene covering one linkage. The great exception that I will highlight a little is, of course, when you put the first GalNAc on to initiate mucin-type glycosylation. If you instead look at the upper part, this is pathway non-specific. This is where in our lab we talk about, this is one of the places where it really snows in glycosylation. This is where the variability lies. This is where you built lactosamine type 1, type 2 chains, so gal beta 1,3, gal beta 1,4, maybe lacdinac. This is where all the silotransferase degrade 2,3,2,6, 2,8, and then ultimately also all the blood group H, ABO, and so forth. And here we have the sulfur transferase. I'm not going to talk about those. But all these steps, the far majority of them, each single step is covered by multiple isoenzymes. This gives you partial redundancy, but more importantly, it gives you possibility for a level of differential regulation. So in other words, you can have 2,3-sialic acid on N-linked glycans, but maybe not on O-linked, and vice versa. And perhaps this is really what our cells are doing in terms of signaling so that it is differentially regulated. What it does is it gives us a very low fidelity in trying to predict what happens if there's deficiency in one of these enzymes. You have so much redundancy, you may overlook it, or you may be basically having false predictions. This is an area we really need to dig into because there is more specificity in these than we know today. So back to the whole scheme. So obviously 2/3 of the genes, we know very well what they do and we can predict to a large extent. This definitely excludes some of these large isoenzyme families, but this is where variability will be. And I'm going to show you now some global information that you can place on this path. So just to sum up for a second, it's 175 genes because since we made this, we have now turned this unknown into a gene called thymine 260, which I'll talk about. Pathway specific, non-specific. I can say that all the known pathways and linkages we have covered by at least one gene that we know will work. Now, I will also show you that we don't know everything. So discoveries are still there. We can define whether linkage is under redundant or non-redundant coverage. That's by isoenzymes. And then we can put global views on what happens, what do we know about the current state of congenital diseases and glycosylation, or disorders of glycosylation, or all these disease traits that we constantly get from GWAS studies or other genome association studies. We can throw those into this system and try to understand how that works. And then I will end up, if we have time, that this also becomes a blueprint for what we can do if we want to custom design glycosylation for therapeutics or understand a biological phenomenon by genetic dissection. So congenital disorders of glycosylation, I'm sure you're aware there are many of these, and Hot Fries, one of the prominent researchers in this field, summarized this a year ago. I think we have a few more now. But basically, you can see how it all started with all the N-glycan deficiencies. Now, I just told you all the N-glycan deficiencies basically are based on single genes covering non-redundantly one linkage, and therefore, and not only that, it's easy to analyze N-glycans, but that is part of this. But we're now seeing, as you can see, expansion. So, of course, GAGs, that's been a long time. But the three forms we want to talk about, O-mannitylation, mucin type O-glycosylation, and O-fuc and O-gluc, they are currently increasing in numbers. And there are some special features we can discuss as we move forward. Over 100 glycosyltransferase genes. Remember, this is a summary of everything, including glycosylation related. But the 174 glycosyltransferase genes, now over 100 of them are associated with a disease with deficiency. These are all very rare. Remember that becomes important as well. The very rare event, most of them. So here's a map where we have put in two sets of information, and it's color-coded with green for known congenital disorder for glycosylation with a phenotype, and then GWAS, which is a bunch of traits, some disease traits, some just traits for, it's not blue color, blue-green eye color or things like that, but traits like low HDL cholesterol, bone mineralization density. And you will see in the literature, all our glycosyltransferase genes are massively covered by GWAS traits. And the researchers that do these GWAS traits, they don't know what they should do when they stumble on a glycosyltransferase, apart from calling a glycobiologist and ask, what could this be? And what should we do? Now, I agree, many of these GWAS traits are probably not causative, probably not involved, but I will give you some examples where they are. What is important to note here, the dark green are genes where there's only a congenital disorder for glycosylation. Now, please note they're all in the N-glycan pathway. They are also in the linker region of the glycosaminoglycans. They are in the whole array of O-mannitylation that leads to the matrix gel glycan important for laminin binding. The light greens, they are where we both have a CDG and a GWAS association. Note these GalNAc transferases here, not very many, and these are very recent, most of them. But note all the yellow, they are all in this area that I said before. This is where the biological information snows. The rest are something we always have. O-man, of course, I talked about, the O-GalNAc, and then the O-fucose, O-glucose. What if we looked at all the data that exists on mouse knockouts? So lethal knockout, those are the ones with dark spot in the middle. They totally superimpose the matriglycan in O-man. Here, the O-GalNAc, mice are fine to a large extent. I remember the first knockout was done on a GalNAc transferase, was done by Jamie Marth's group, one of the most experienced mouse geneticists, immunologists that has taken out glycosyltransferases. And his paper was basically no phenotype, a lot of work, and this must be because there's functional redundancy. Of course, 10 years later, they reinvestigated the mouse and found out that it had a subtle immunological phenotype. But you can live with individual ones of these GalNAc transferases, but you can't not enough with others. And then the mouse knockout, see many out here and very faint or subtle phenotypes. Bob Heltinger will tell you more about that. Now, if we then superimpose, and this is not preliminary data, but it is preliminary in the sense that this is based on organ transcript data, okay? So if you, on a scale, take 10 or 15 organs and you ask which genes are differentially expressed, and you put a scale to that, then blue means not really changed between 10, 15 organs. Red means highly changed. And what happens? No regulation in the N-glycan. And I'm sure you know why we need it. No regulation, so we can do this. It's a little difficult with this double, but here you see very little organ differential. I mean, this is just transcript level. It's not yes, no. It's the intensity of the transcript levels. O-GalNAc, massive changes. O-fucose, somewhere in between, but you can clearly see this. But again, when you look up here, this is where it snows. Now, obviously, we need to move to single-cell transcriptomic to fully capture the implications of this. But what it tells you is that it may be that, apart from the carrier proteins here, the capacity of making certain types of glycosylation is there, but we vary the branching, the elongation, and of course the capping, mainly of sialic acid, to drive biological interactions. And we need to go to single cell in order to identify true regulatory hotspots. And there's a lot of work to do, and I can show this without really saying much more, except that here in Joshi and Liu Dworking last year published our first paper in this field. And it's basically a methodological paper for how can we extract single-cell transcriptome data. And there's a lot of issues with that, because it turns out that the glycosyltransferase genes are lowly expressed. And so a lot of the single-cell data today, but not tomorrow, are of insufficient sensitivity to grasp and quantify significant changes. But we can for certain things, and in several cases, including some of these, actually they were able to demonstrate what we already know. Changes from in sialylation, or changes in these two GalNAc transferases, or MGAT3 in glycan. So this is a really important field to move. And what it will do, it will move us from analyzing glycans of a homogenate of a whole arm or a liver or a kidney to actually, that made you wake up, to actually look at the single cell and what it does to change its glycosylation machinery to accommodate particular functions. So in other words, this brings us to single cell glycomics although it would be predictive of the glycosylation capacity. It won't show the actual glycans, but we need to correlate prediction of glycosylation capacity with the actual glycans. So this was our little introduction that was about 20 minutes, and now we quickly switch to only talking about my favorite, and that is the mucin type, GalNAc type glycosylation. This is obviously the most important type of protein glycosylation that you will hear about until Bob starts a little later after the break. Why is it important? Now, mucin type, this is mucin type, no? You basically have 5,000 amino acids, and every third amino acid has an O-glycan on it. Now, that's a lot of sugar. But there are other proteins that have very discrete positions of O-glycans, and those may be really interesting to study. And so I will dig a little into that, and we're using this framework. I stole that from you, so now we can see we're talking about this. We're talking about 20 isoforms. We're talking about a GalNAc alpha linkage to serine threonine, and also tyrosine. So we've found a number of tyrosine sites that are O-glycosylated, including one on the amyloid beta protein. A very interesting place, which Adnan Halim published some years ago in PNS. We still don't know what it means. And we don't know much about the stoichiometry, and we don't know what enzymes do this. But we've now made a cell that has no GalNAc transferases at all. It's alive, it's happy, it can secrete proteins. It's just a cell. But still, surprising to me that you can eliminate all of them, and we do not see any tyrosine GalNAc glycans either. So I'm pretty sure, although I have no direct evidence, that all 3 amino acids are covered by GalNAc transferases. So, again, we have the pathway specific, and what is important is that all steps, maybe apart from the core 2, in the elongation, so the next step, are covered by single genes. But you need 20, let me just, I'll get to that later, but in fact, we only know like 14, 15 of these are truly active. So some could be pseudogene, but from sequence, they look totally perfect. But you know, there's a lot of redundancy and potential for differential regulation of O-glycan positions. So think about perhaps a protein is a protein when it's expressed on one cell, but an O-glycoprotein when it's expressed on another cell. That's the possibility. Now that is not a possibility with most other types, at least N-glycosylation. There, a protein will always have N-glycans at the positions. And then these core extensions, which is kind of interesting. There's another interesting feature that Rick Cummings' group has sort of pioneered, and that is that the first step, the most common step, which is a gal beta 1,3 GalNAc core 1 synthesis, is made by an enzyme that has an obligate private chaperone. So if you knock the actual enzyme out or its chaperone, you get the same effect, because the chaperone is required to make the enzyme fold and get out of ER into Golgi where it should function. If we look at the extension, then, as I mentioned, the elongation, branching, and capping, those are done by the same enzymes that do N-glycans and glycolipids. And then we have all these isoenzyme families, and it becomes very difficult to predict what actually goes on. Now, clearly, we are beginning to, well, we already know that ST3-gal, where do we have ST3-gal 1 and 2 work on the core 1 O-glycan here, but we really need to validate that in cell systems that there's not others, and in fact we are seeing there are more. So A little historical background, because now I just throw out 174 5 genes and say this is the glycome. You can just use that. How do we get there? That's not an uninteresting sort of avenue. Both Bob and I were in the field when you purified enzymes. You know, you took a whole liver and you extracted enzyme protein. You spent 3 weeks and then the enzyme was dead. Then you did it again and you did that for about 4 or 5 years. And finally you had 5 microgram of protein and then your amino acid sequenced it. Then came the transfection cloning, where you throw a cDNA library into a cell that misses a glycan phenotype that you can select for. And then you could clone that. That worked really well for a few genes. But then came the human genome data in the 90s and up until beginning of 2000s. That's where our field really blew. And that's why, importantly, we can say we probably know all the glycosyltransferase genes that are similar to the ones we know, no? So we know there are 20 GalNAc transferases, even though, because they look alike. And every time we find a new one, which I'll show a few examples of, they are different. So we are pretty close to that. And of course now we can use gene dissection and basically toy with all this. Now the next level, well, this is my favorite historical. Let me just give you an example. So this started with Ed McGuire, that in famous Saul Roseman's lab that has trained most enzymologists in the field. In '67, they demonstrated the first enzyme that adds GalNAc to a mucin. That was the beginning of mucin type or O glycosylation. I didn't copy it, but at the end of the text, they suggested that this enzyme may do other types of glycoproteins. That's how little we knew about how many enzymes there could be. It took many years for people to get to it, and this is about a decade or more of my life where, thank you, Cole, 2 dentists. Now remember, Larry Tabak, the director of NIH, is also a dentist. Here, we every year met at conferences with the new GalNAc transferases until we ended up to about 2. So my name is Bennett. Bennett was the lead guy in our lab. And then about 2002, then Yoshiki Narimatsu, who you will see on Friday give a talk, his father took over because at that time, both Larry Tabak and I had run out of favors with the funding agencies. The funding agencies didn't think it was funny to clone another enzyme. I actually have a rejection letter from one of my grants that says, we'll give you $100,000 if you don't tell us about this anymore. And go away. They are, of course, wrong, because this is really where it's important. Anyway, we had fantastic time. And Larry Tabak is the most funny guy. And yeah. Okay, now, how do we then take all this prediction and get closer to what is really correct? Now we need to define the functions of these enzymes. And a little like we discussed, or Matthew discussed, we know a lot about how sugars interact with proteins. In this case, we know how simple sugars, or for some of them, that would be peptides, how the enzymes in a test tube work. But that does not, it gives us very good hints, but it does not explain the full repertoire. There can be other things in, and that's where in cells where you use overexpression, in the old days, now knockout, knock in, and you do it in a combinatorial way, then you can actually, in cell systems, in tissue models, of course, in whole organisms you can. It's a little difficult, but we're learning a lot from the CDGs I talked about. But in fish or in Drosophila or other, in mice it's a little difficult to do massive combinatorial, but in cells we can, and there we can learn. And I think I would just highlight Hans Wandel from our center that is doing this in a tissue model. So it's in one cell, but a cell you can differentiate in skin, and he's now developing also a neural model where you can ask individual, here I show where he basically did what we call the glycoconjugate library. So he took out information about glycolipid, all the information about elongated N-glycans, all the information about elongated O-glycans, and of course also on O-fucose. And then you get a phenotype. Now he's now done it for the GalNAc transraces. In this paper, I'm not going to show a picture, but I suggest you look at this. This gives you a more advanced readout than in cells, of course. The other thing we need to do is we need to define what is the glycoproteome. Where are these sugars? And when we started out, there were obstacles. We knew a lot. We knew maybe 100 proteins that had O-glycans on because they'd been isolated and digested and so forth. But we didn't know where are all the O-glycans. We didn't know how, whether it was mainly mucins or was also on other proteins. And the key, of course, is that we don't have an enzyme like PNGSF that's used for N-glycoprotein. When you use PNGF, you leave a scar, basically an amidation of the asparagine. And then you can say, well, there's N-glycans, and that's eminable for mass spec. So if you leave the glycan with its heterogeneity on, then each glycopeptide doesn't become 1, but maybe 10 or 20. And the masses are different, and it's totally difficult to identify. So we developed the simple cell strategy, where we combined engineering. And if you're interested then in mass spectrometry, then Sergey in our lab has then developed that further. So we are beginning to be able to do glycoproteomics on more mature sugars. But let's discuss the simple cell strategy. And this is based on Rick Cummings' work, that if you remove COSMIC, then you basically truncate the O-glycans by inactivating the C1-GLT1 enzyme. It actually doesn't matter whether you take this one or this one. We sometimes do, well, this is an early slide, so we use this. Later, now we're using C1-GLT1. But then basically everything goes down to TN or sialyl-TN, and sialyl-TN is a sialic acid, easy to remove with neuraminidase. COR3, as you can see here, is restricted to certain or to GI tract, and it's almost never expressed in cell lines where we use this strategy. So it's not really a problem. And here you see the validation. So you see wild type cells, they have no TN sialyl-TN. You take out simple and there. And here you see the enzyme, it's gone. And here you see the elongated structure, and it's gone. We've done that on many, many different human cell lines, and then basically had a workflow where you can capture all these glycopeptides after neuraminidase on a very long, 2-meter long lectin column, VVA, or it can be Jacqueline. And then, of course, you can do high fractionation, and you can do standard mass spectrometry sequencing. And now you have a SCAR, or you have the GalNAc, so you know, and you're missing, though, occupancy, of course, and it's really sensitive. So it's one problem that we may actually overpredict. So what comes out of this? This is an old slide, but I refer you to the glycodomain viewer, where you can put any protein or gene in and get the latest update, not only of our data, but all published data on the O-glycoproteome, and actually puts in N-glycans also. But what we learned was that almost every protein is O-glycosylated. And they're O-glycosylated, in mucin, like stem regions, so in clusters. There's some single sites ER proteins are O-glycosylated because they traffic the cis Golgi. There's these long stretches, like almost mucin-like, and the sum of it is that I usually say 85, but most protein going through the ER, they are O-glycosylated. We have more than 3,500 human O-glycoproteins, more than 15 sites, and O-glycans are mainly in unstructured linker stem regions between domains. It makes sense because it's initiated in cis Golgi. The joke about all our data is we never see mucins, and we never see, or rarely see, the juxtamembrane regions, and it's obviously because of capabilities to digest and get a certain length glycopeptide or peptide that you can get out. So there are ways to get around that, and we can get to that. So then we can talk a little about the functions. So O-glycans basically exert or participate in most of the functions that we assign to glycans. And the typical ones, the general functions, we obviously all know, and they were talked about earlier. But I wanted to point out some specific functions where glycosylation or the glycan becomes really a driver of protein functions. And, regulated protein processing at the surface, ectodomain shedding, we have very nice examples. Ligand binding, I will give an example. Interesting that I think nobody thought about was that protein processing could be regulated by O-glycylation, and I'll give an example of that. But other than that, obviously, O-glycans have the capability of presenting very high density of sugars, but also unique patterns, and I will discuss a little more about that. So here's the first example. It's some years ago. Out of the blue, an Israeli group led by Topaz published that GalNAc T3 led to familial tumoral calcinosis. Nobody paid much attention to it. It's like I said in the discussion, it says it's some glycosylation gene, we have no clue how it works. So I read that paper, and it was about the time when they when we started our center, and it said that, strangely enough, FGF23, which is important for familial tumoral carcinosis, was highly upregulated, 24 in patient. Turns out as a compensatory because it doesn't work. The problem with FGF23 is that it needs to be cleaved. The full length is active, but if you have too much of the full length, you get hypophosphatemia, basically Ricketts disease. If you have too little, you get hyperphosphatemia. And there's a furin inactivation site there that's important. FGF23 works in the whole circuit of phosphate reuptake, and it basically block kidney phosphate reuptake, and that's how it works. So I looked at the sequence, it's RXXR, but in this case it's RHTR. There's a threonine. Is that possible? That's the luckiest day in my life. We threw 20 enzymes, the enzymes we had on it, and only GalNAc T3 can put an O-glycan on this site. That's the unique function. Later, an American group, I forget the last name, but Tagliabrati, demonstrated that also extracellular phosphorylation by FAM20C is important. And basically, you have a crosstalk between phosphorylation of serine, which blocks O-glycylation, and O-glycosylation, which blocks furin. Those three is a knot where three quite common types of of post-translational modification hits and makes it very specific to regulate only that particular growth factor. Then you can say, well, is that important? There's, I think, several hundred, so it's not rare as CDGs, but it is a rare disease. Now, what is important is this sentence. Later, in the beginning, it was only GalNAc-T3 that was identified, but later, about 20 or 30% of all these patients were shown to have FGF23 defects. So here you have an enzyme, GalNAc-T3. I know a lot about that enzyme because we originally cloned it. It glycosylates almost everything you give it in terms of peptide. It clearly is important in mucin glycosylation, many different. But the only thing it does of, in this particular set, it's in a human, only thing it does is exactly the same as FGF23 because the phenotypes are clinically indistinguishable. I think that tells you something about the power of isoenzymes and regulation of particular things. Now, why is that interesting? Well, it's a little interesting because when you screen for bone mineral density, then you pick up GalNAc-T3. So GalNAc-T3 is central for the circuitry of our serum phosphate and our bone mineralization. And how that works, we don't know, but it has to be regulatory. So there will be a different regulation of GalNAc-T3 because obviously it's there. Another example that came out of our simple cell approach was O-glycosylation on LDL and other LRP receptors. Now, I alluded in the very first picture, and I don't want to go back, but everybody knows that there's this mucin domain in many of them in the stem region. So it's known that LDLR needs O-glycans. What was not known is that in between these ligand binding domains, which are very close to EGF repeats, actually an EF-like repeat will treat disulfides, there's a 4 amino acid linker in between these, and that goes for all the receptors. Remember that LRP2 is megalin, LRP8 is ApoER receptor. So these O-glycans were not found, and I forget to mention people, but this was Shenyang Wang and Katrine Scholler that has worked on this for many years. Now, what is important is that just looking at LDLR, if you just take LDLR, it has 4 of these linker regions where there's an O-glycan. If you knock out T11, which I won't show you the data, then they disappear. Nothing else disappears in the O-glycoprotein. We have very nice quantitative. It's only those. If you put GalNAc-T11 in, that's in this paper, if you put GalNAc-T11 in an inducible form into a cell where you've knocked it out, a liver cell, HepG2, then you see a linear response curve on the glycans and this capability. What I'm showing here is uptake of fluorescent LDL. Please note that this is wild type. This is a GalNAc-T2 knockout, just another family member. Nothing happens. This is knockout of LDL, so the receptor by itself. There's always background in this assay. And this is when you take out GalNAc-T11. It's very dramatic. There's an affinity, a 5-fold decrease in affinity for the ligand. And if you then take out cosmic instead, now that will affect all the glycosylation, but you get the same effect. And that means it's not the GalNAc per se, but its ability to carry acylic acid in core 1. And maybe the acylic acid is important in the binding, or maybe the acylic acid is important in bending. So basically, it's a structural problem, a fold problem. As you may know, these receptors, they go between bent and open conformations, and maybe that's what drives it. Now, why is that important? Well, so together with Larry Tabak, actually it is Kelly Ten Hagen, but Larry Tabak is there also, they had a mouse, and we looked, and yes, sure enough, you basically have protein in urine. This was all only focused on kidney, where megalin is the major reabsorption mechanism for protein. If you remove megalin, the mouse locally in kidney, the mouse will excrete protein in urine. If you remove T11, it excretes protein, and within some time, the kidney basically undergoes fibrosis and doesn't work. Important, but then again, genome-wide association, T11, I'm sorry you can't see the whole thing, but down here it says GalNAc T11. It's a chief as candidate for kidney function decline. And again, it has to be regulatory, it's not structural. So it is a deficient regulation that drives that. Okay, so so I got lost that you didn't turn on the time. It will ring 5 minutes before. It said the same number a long time. Last, if GalNAc type oglycine is interesting in cancer, why is it interesting? Well, if you look and sort of what changes do we really know that are consistent in cancer? There's a lot, and I put some arrows in N-glycan, of course in the GAGs, and of course there's these in the non-specific pathways with sialyl X, sialyl A, and things like that. But if you really look at it, the most prominent is really truncated O-glycans. And it is the expression of these T, Tn, and sialyl Tn. And this is kind of interesting. Number one, you all have autoantibodies, most IgM, but for sialyl Tn it's actually IgG, to these structures. It's very easy to develop antibodies to them in mice, so there's no tolerance. And you can also get very potent antibodies, which I'll discuss in a second. So here's the pathway, and these basically, you downregulate all these, or there's other things in the cell, which I'll discuss. But in principle, you end up with Tn, sialyl Tn, or COR1, and those are, a long time ago, they were defined as pan-carcinoma antigens. I mean, they are really prominent, and the interesting thing, of course, is we have autoantibody against them, which means, which put high validity to that statement. Here's just an example. I've used it for now more than 20 years. Well, 15. But basically, if you ask the most expressed mucin, actually in most tissues, but in particular in the breast, MUC1. The mucin MUC1 is a large mucin. It has these variable, but maybe 100 tandem repeats of 20 amino acids that looks like this. And there's these two glycans here. If you have an antibody that binds MUC1, it looks like this. In normal, it's very nicely oriented in the apical membrane, and in tumor, it's massively overexpressed. If you generate antibodies that can only see Tn, but funnily enough, see them in context only with MUC1. These are antibodies that you can elicit in patients because we have immunized 20 people or patients with this, and they get exactly the same antibodies as this 5E5. And you can also screen for autoantibodies, so spontaneously 20% of cancer patients will have antibodies to this epitope. You can clearly see it's not expressed in normal, and it's expressed widely in the tumor. So these we call glycoproteoform specific epitopes or antibodies. Now, why is it we get there? There are in the literature a number of explanations. I would love if it was simply the glycosyltransferase expression, because that's obviously what I've been talking about now for who knows how long, 40 minutes, 30 minutes, that it's the enzymes that drive it. But clearly there's general disorganization of Golgi. Frederick Barth, who's now in France, but was in Singapore, has actually demonstrated that GalNAc Ts, maybe under stress or other SARC-induced conditions, can move into the ER And then they will prematurely put sugars on. And that could be why you get it. And then, of course, there's Rick Cummings that has found mutations in COSMAC. They're very rare. We have checked. We've checked a large cohort of pancreatic cancers, and mutations in glycosyltransferases are very rare. Also in the normal population. Loss of heterozygosity, most likely hypermethylation. So it's probably a mixture of everything, but there's not a linear and easy explanation. But you see them almost always. So very shortly, I will introduce the OMAN glycosylation, but I'll do that mainly from a discovery point, because Monday morning, Edna and Halim will have a nice presentation of this. So now we are here, and you can see we have 3 different types of O-mannose. They're basically the same. It's an alpha mannose to serine threonine, and there are 3 different enzyme families or single enzymes that direct them. Background for this, and you may have seen we had a snapshot some time ago, because we were sort of wondering of the evolutionary sort of origin of glycosylation. And you know, the yeast O-mannose. So in yeast, the only type of mannose is O-glycosylation is O-mannose, and it's done by 7 or up to 7 PMTs. These PMTs are homologous to POMTs that is, I can't see, but maybe this one, that are the classical O-mannose that were found later in, actually quite recently, I mean 20, 25 years ago, where we found that O-mannose was also found. Now what was interesting is that we analyzed yeast O-mannose, and the O-man were positioned in proteins very similar to how we in the human protein, even on some proteins that were homologous, in the same regions as O-GalNAc. So we thought the PMTs, or the POMTs in human, would also only glycosylate, so would glycosylate quite widely. Now that turns out not to be the case. So this is the POMTs. Now remember, POMTs, they have O-mannose that goes on alpha-dystroglycan and makes this fantastic, complicated linker region to end up with the glycan, glycosaminoglycan-like structure. POMTs, as a summary, they serve really only alpha-dystroglycan and very few proteins, like one that's called KIA 1549. We don't know what it does, but it's also like a mucin protein. But what was interesting, well, obviously this is the glycan chain, and you can see laminin binding to it, and you can see it's hooked up on this, but then you have O-mannose glycans that are not. They simply have the glucanac and then a galactose and a galactose, maybe a sialic acid, and you also have O-glycans because it is a mucin-like region, but they're spaced. But we asked, with our simple cell approach, what is the O-man in human? And very quickly we identified not only alpha-dystroglycan, of course, but also this protein, which I won't talk more about, but then plexins and cadherins and protocadherins. And just to give you a feel for the cadherins, here's all the cadherins, type 1 and type 2 chain, and here's this evolutionary in E-cadherin and cadherin 11. You can see that these are very conserved throughout. So then we asked, who did all that? It must be POMT1, POMT2, and there must be some specificity in those. So we took out POMT1 to POMT2, did a differential labeling, the same flow, but instead of using VVA, we used CONA, and then we measured. And here's a surprising result. We took out POMT1 alone, POMT1, POMT2, POMT1, POMT2, with or without simple cell, and the only thing that disappeared was alpha-dystroglycan. The cadherins and the protocadherins stayed, still O-mencylated. Many years after, here in Jersey, we had all kinds of bets for what the enzymes were. He won all the beers in the lab. We have knocked out maybe 10 other candidate genes before we came to the TMTCs, 1 through 4, does cadherence, and TME 260 does plexins. And so now you have 3 different, and you can see they have the same GTC fold, but they're very different. You cannot identify them by sequence similarity, and the TMTCs have these TPR domains similar to OGT. They're not elongated. We didn't need to use our simple cell approach, which kind of, I mean, it's fine, but a little depressive. We spent tons of years making the cell lines for this. Not necessary. In contrast to this, now if somebody wants something to do, we need to figure out why are these only on some sites, and why are these not elongated, and what are the functions of alpha manos out there. Now, the only thing we can say today is that both are members of CDGs now. So the TMTCs, there was a paper before we knew what it was on cobblestone lysensiphy, and also on the TMT 260, there's a rare syndrome with cardiac effect and multisystemic disorder. So what are the TMTC? That was the isoenzyme I talked about. Well, they actually not isoenzymes per se, because they seem to work on different sides of the EC domain, which is different beta strands, and some glycosylate on B and some on G. And Monday at 9, Adnan will tell you, because this is unpublished data, a lot about this, I'm sure. Why are we on time? Because it doesn't say. Okay, I'll quickly close your eyes for a second. It's only 10 slides. It looks like a lot. Don't worry. Take-home messages. You need a global view. I wanted to show you that you can do cell-based glycan arrays, but Yoshiki Narimatsu and Tabacon that sits up there, they will Friday, for those that don't fly out early, give talks on this. So it's no necessary to repeat, but please go and listen, because there you will see that you can actually present sugars on proteins in any way you want, and with any types of glycosylation. And then I just want to time all our people. And then I want to highlight that we have a number of stipends, PhD stipends, if they're good master students or postdocs available. Thank you.
Thank you very much. We have time for questions. And if nobody asks questions, you can continue with your slide.
That's a good thing. That's a good
Okay.
idea. No.
Anyone with any questions for Henry?
It's not a very active.
Very nice talk. So the novel O-mannitosyltransferases, you couldn't pick them out by sequence. So what do you think the evolution of them?
Well, they're the same domain structure, but yeah, they're very far. Yeah, I don't know. To be quite frank, you can, now when you know, you can see that the first luminal loop is the catalytic unit side. And that's for POMTs and also for these. And you can see if you put amino acid and you move a little yourself, you can see four or five amino acids. But even the catalytic amino acid is not clear.
So they don't have a catalytic center, basically.
No.
Just to follow that up, sir. You mentioned that you have 175 genes. So do you predict that at one day we discover something entirely new, like we discover alpha-dystroglycan with all these genes that we never know before, or with this ominosyllate pathway? Would there more surprise waiting for us?
Oh, I'm sure.
So
I'm
you-
sure. And the way to do it is, and I think it's so illustrative, we went into in a boring project where we just wanted to know where's old man. And then you find out that there's all kinds of places.
Right.
And you get new classes. And if you think about cadherin fold domain is very different. The IG domain or plexin transcription factor domain is called the IPT domain in plexins are very different. And then you had this unstructured region, and it seemed unlikely that it could be the same enzyme. And then you look at it, and it's totally different.
Right.
So by shutting off something you know, you may find surprises. I haven't found more yet, but
For the cadherin repeats, do they need to be folded to be modified, or do they get modified before they fold?
yes. All I can say is that we can express beautifully secreted cadherin modules or ectodomains of E-cadherin without OMAN. And we can also get them E-cadherin to the surface. We're still arguing whether there's a difference or not. And we've not been able, that's why I don't go into the function, but clearly they are functional because otherwise you wouldn't have CDG. But we have no clue exactly how they work. The E-cadherin, I didn't mention that, but the E-cadherins is not EC1. And remember, the cadherins have EC1 trans interactions like that. And then the OMANs are here. And for the lattice to be formed to make strong, you need to coordinate them in 2 dimensions on the surface, and then you need this. So I favor that the mannoses coordinate the lattice. But then the protocadherins, they do 3 EC1, 2, 3 trans. But they have OMAN all over. So we don't know.
Cool.
I have a question about the guarantee 3. And so you said that guarantee 3, just only one enzyme can synthesize the O-glycosylation of the FGF23. I forgot the threonine 173. And so why only guarantee 3 synthesize at this site? And is it a structural problem or
Yeah,
tissue specificity,
Yes.
expression, or which one?
Yeah. So for sake of time, I shortened it a little. So some of these GalNAc transfers come in subfamilies. And there's a GalNAc T3, T6. They have almost identical substrate specificity. So T6 can do it in vitro, but it's not expressed
Not
in
expressed.
bone marrow where T3 on osteoblast and class that does this. Now, we have, I think I cited, but we have a paper with Ramon Hurtado Guerrero, Spanish structure. So we have a co-crystal of GalNAc T3 with the FGF23, and it's totally fantastic. So GalNAc T3, 178 is where you want to put the sugar on. RHTR phosphorylate. 171 has an O-glycan. GalNAc T3 docks the lectin domain, which Maureen talked about. Or was it? I forget. One of our lectin speakers.
Okay.
Docks in here, and the catalytic unit then docks in 178. If you remove the lectin domain of T3, it cannot glycosylate.
Oh, okay.
So it's a very regulated, and that's captured in the structure.
So lectin domain is important also.
Lectin domain is important, and T3 it has low kinetic
Yeah, yeah,
efficiency
yeah,
for
yeah.
this site.
It is very interesting. So,
Yes.
excited. Thank you very much.
You talk about the importance of glycotransferase, but how about the enzyme for hydrolysis? Do you need to know that in order to know all the glycosylation?
That's a very good question. And up until recently, I would probably have said you don't need to know a lot about the hydrolases because it's a lysosomal. So in human, we don't all the lysosomal hydrolases. But if you heard, although I didn't because I came so late on the plane yesterday that I died, but I would have liked to go to Ludger Johannes' talk yesterday at 6 o'clock. Did you hear? Then you will know that the neuraminases that are lysosomal, the new 1, 2, 3, 4, there's only one that's lysosomal, but the other ones, they can be in the membrane. And they can be triggered by pH change to change of remove sialic acids. And a lot of stuff can then happen. So I think you need to include the neuraminases that we have in this equation. I don't think it matters a lot for what we do here in terms of genetic dissection and understanding the pathway. But there's no major remodeling going on.
How about the fucositis?
I don't think so. Do you know something I don't know?
Got you.
It's absolutely possible. But we're not talking about in plasma, we're talking about in the membrane or in the secretory pathway.
Right.
Yeah.
Any more questions? I would like to return to the O-mannose. It's really fascinating to me. So, did I hear correctly that all the one that is elongated by TMC pathway, the O-mannose are not elongated? They are single O-mannose.
Well, as far as we know.
So only the POM-MT mediated one would be elongated.
Yes.
Okay.
Somebody needs to figure out how that works. But obviously, the O-mannoses in alpha-dystroglycan and also in this KIA protein, they are in unstructured or fairly unstructured regions.
Yeah.
These are, maybe not totally buried in the fold. There's actually an interesting thing was the guru in coherent structure, Larry Shapiro, had seen something that looked like a hexose buried in his crystal structure, but ignored it. But we found, when we found out, we collaborated with him, and we found in his supplementary figure 115 that you actually could see that it was a mannose, and they were buried in the structure. So the question is whether they're accessible or not. But you couldn't really tell. But that could be one.
I have another question about the O-mannose transferase. You got the 5 new O-mannose transferase. Is it exist in the Drosophila or also in the worm?
TMAIM 260 is the oldest one,
Okay.
and perhaps the founder.
Okay.
TMTC, so and TMTCs are also in Drosophila, where Vlad Panin came up before. He is looking at them and is seeing some very interesting stuff. Axon guidance, but we haven't been able. I mean, he should tell him himself. It is not published,
Okay. Thank you.
but definitely interesting.
When the PPN Gautiens is the model organism a good think to study their specificity? We talk about the glycobinder protein. There's difference in mouse and human, etc. So, would you use mouse or zebrafish or C. elegans or whatever to study the GalNAc transferases?
If I were putting my money on it, I would go with GVAS studies. I would use GVAS indications. There's GVAS candidates for all of them almost, and I would follow those. Since we have time, we made a cell. So remember, when you have a cell, it will have 5, 7 GalNAc transferases. There's only one cell in the body that only has one. That's the sperm cell. It only has T3. Every other cell has many. So we study their function by knocking one out and then do differential. And we have 3, 4 manuscripts lying that we just can't get, but it's too complicated. Then we did another thing. We made a cell that has no GalNAc transferases. I think it's the brightest idea we've had in a long time. No GalNAc transferase, and then we put all of them in one by one. And then we compared the glycoprotein. What do you do? So we put T1, T2, T3, which they're basically indistinguishable glycoprotein about the T3 and FGF23. Massive. Everything is glycosylated. And we put T11 in that only does it regulates your endocytic receptor functions. Almost indistinguishable, a little less intensity, but the same. Then we put the ones, I thought that was the brightest, the ones that nobody has seen any function for. It's called Y family. They are in the ER. Nothing. They do nothing. We probably did something wrong. But I just think that it's too difficult. We have 4 different rats going around with loss of 4 GalNAc transferases. And the only one we are confident about is T11. It's a mouse, but we also have a rat, where we can see the LDLR receptors. All the other ones, you gotta remember, if it only is one protein, I didn't tell you that, but we haven't seen FGF23 in any glycoproteomic screen. It's secreted, very low amount. It's very potent. You don't see them. So the chance that you see what it is it's doing because of proteolytic digestion, because of your cell systems, the organ you're looking at, it's tough. We need to learn, but I think you need a hint. And that also is better funding-wise. If you're pursuing osteoporosis, it's better. Just an old man's advice.
Maybe one quick one. Since I think you had it on 2 slides, so I can't help to ask. But a nice example, you were saying, if you look in a simple in vitro system, 2 enzymes can do the same thing. The example that you had was ST3-gal1 and 2. Yes. So Jim's old literature said both hit GalNAc beta-1 3-gal.
Gal beta-1 3-gal.
Yeah.
Yes.
I said that opposite.
That's fine.
But I haven't seen in cells where ST3-gal2 is doing ST3-gal1 work, which is sialylating core 1. Is that definitively happening in cells?
Yeah. So if you're here Friday, take a dance with Yoshiki, but I'll give you this shot. If you take out ST3-gal1 and 2, you don't necessarily remove all 3 glycosylation on core 1. You need also 3. Not in all cells. If you take out 1, you still have this. You need 1 and 2. But that's based on core 1. If you then, you refer to Christian Bühl's paper, if you then put them back in, then you learn something different. ST3-gal1 can do core 1 and core 2. Remember, core 2 has the gal. It can do both. ST3-gal1 can only do core 1, cannot do the core 1 arm in core 2. So there's some really specific subtle differences that you find.
Any more questions for Henrik? If not, I got to ask the last question. Well, we used to be taught like Yuka Fine and Tenfei, they work on these old manuals before, and they characterize the first structure, etc.
Kids and
And I have
sulfate.
this idea that old manuals is very prominent in brain.
Yes.
So is it still true now, you think, that why brains selectively use old manuals for glycosylation?
Well, so the protocoherence you obviously have. But the funny thing is that I mean, you know, Tenfei's old release sugar data. I don't know whether it's because of the amount of protein, so that you have more mannose because you have more amount of the proteins, or they're different proteins. But when we do a brain glycoprotein, which has been done, although the sensitivity is a little problematic, but then there's nothing particular that pops up except cadherence and protocoherence and alpha-dystroglycan. So exactly what lies behind it. But your question, I think, relies, are there 4 new types of proteins that happen? No. At least we don't know.
All right. Okay.
With the caveat that they are elongated in the brain. Because
Okay.
we haven't been able to make a simple cell human or mouse yet,
Right.
as you know.
Right.
So
Right.
with that caveat.
Well, we do find that the old manuals, the elongated old manuals, are more abundant in.
Oh,
in
of
brain.
course. But that comes from alpha-dystroglycan.
Okay, I think we have a very fascinating talk by Henrik, and hopefully by now you'll be fascinated by the world of O glycosylation. And we have a short coffee break, and then come back with more O glycosylation. Okay, we'll be back in 20 minutes or so. Thank you. Earlier, which with me introducing Bob, so that Bob would have all the time he needs to go on. Okay, so please be seated, and we'll start the next session. Okay. So, as you heard before, that Bob is going to tell you about this another fascinating aspect of O glycosylation. So let me use a little bit of time to introduce Bob. Robert Hortiwanger, and I think the first thing he did with Robert Hill at Duke University, where he started with purifying glycan-binding protein, lectin. And then he switched to go to glycan instead of glycan-binding protein. And he joined Jerry Hart's lab, which I think everybody knows Jerry Hart, for his discovery of O-gluconate, whereby in the Jerry Hart's lab, he purified and characterized the O-gluconate transferases. And then he moved on to Stony Brook University, and for his first independent appointment, and he stayed there for more than 20 years, I think. Yeah, okay. And then at that time that he discovered what he's going to tell you today. I remember that when the paper came out in Nature about this fringe and notch, it was such an excitement at that time. Okay? And ever since then, he and Pam Stanley and a lot of other people have advanced the field so much that we reach where we are today. And another thing that you know about Bob is probably also that he took over from Ron Chanel, who took over from Jerry Hart as the editor, chief editor of glycobiology. Okay? And he served there for 10 years, right? And just recently he stepped down. So it's our pleasure and our privilege to have Bob here to tell us about this fascinating world of O-gluc and O-fucose. Okay, Bob.
Can you hear me? Good, great. So thanks, Ku, for the introduction and for the invitation. It is great to be back in Taiwan. And today I'll be talking about O-glycosylation of these cysteine-rich domains. These modifications are much less frequent than the modifications that Henrik talked about, but they have very important biological functions. And so I hope to convince you by the end of this lecture how important they are. So there are two cysteine-rich domains that I'm going to be talking about. One is the epidermal growth factor-like repeats, and the other one is called, which I'll be abbreviating EGF repeats. And the other is thrombospondin type 1 repeats, which I'll be using TSRs. As I mentioned before, there are actually several dozen types of cysteine-rich repeats that exist in nature. But we study these because they have unique carbohydrate modifications on them. And so what I'm going to do is, for each of these repeats, I'll define sort of what is an EGF repeat, what is a TSR, and then talk about the modifications that occur on those modules and the biosynthesis of that, and some of the biology that we know exists that associate with these different domains. And then some common features at the end that the O-glycans, how they function, the mechanism of what they're doing for the proteins that they modify. So we'll start with structures and biosynthesis of EGF repeat O-glycans. So just to define what is an EGF repeat, these are small protein domains. They're about 30 to 50 amino acids in length. They have 6 conserved cysteines forming 3 disulfide bonds in a very specific pattern, 1, 3, 2, 4, 5, 6. And those disulfide bonds constrain the 40 or 50 amino acids into a 3-dimensional fold that looks like epidermal growth factor. So that's why they're called EGF-like repeats. The other amino acids in the EGF repeats can be quite distant. And so they're defined by the presence of these 6 cysteines. You can actually, just based on the spacing of the cysteines in the primary sequence, you can identify an EGF repeat. So if you go on UniProt, there are literally hundreds of proteins that have these, and again, they don't have structures of all those proteins, but based on the spacing of the cysteines, they can identify where EGF repeats are. And again, they often occur as non-identical repeating domains in proteins. So incredibly, at least to me, there are 4 distinct types of glycans that exist that have been identified on EGF repeats. Each one, the colored amino acids are where the glycans go, and there are consensus sequences for each one of these. And so I'm going to go through these sequentially, sort of going from the amino terminus to the carboxy terminus of an EGF repeat. So the first modification occurs between the conserved cysteines 1 and 2 at a sequence X-seer, X-proline, alanine, cysteine. And if we see this sequence in the context of an EGF repeat, we can predict pretty accurately that it's going to be modified on a serine. And again, this is unique to serine, not a threonine modification. But with that serine is going to be modified with a glucose and a beta linkage. And so this is the glycan structures that can exist at that site. The enzyme that adds the glucose to the hydroxyl of a serine is called protein-O-glucosyltransferase 1, or POGLYT1, and it's a beta-linked glucose to the hydroxyl of a serine. The glucose can actually be elongated with 2 xylose residues catalyzed. There's 2 enzymes that catalyze the addition of the first xylose, glucoside xylosyltransferases 1 and 2. And then there's 1 enzyme that will cap that, called the xyloside xylosyltransferases. And Hans Bacher group identified these enzymes, I think, 2010, 2011, something like that. Yeah, it was a while ago. Yeah, so it's interesting that the POGLYT1 can also transfer xylose to some EGF repeats to make an O-xylose, a beta-linked O-xylose there. And this is probably because of the similarity of the structure of xylose and glucose. The xylose is a pentose, but it's usually in the pyranose form, and it's missing the extracyclic carbon methoxy group. And so you could see how this, when they're charged on UDP, because they're both UDP galactose, UDP glucose or UDP xylose are the nucleotide sugars for the transferase, how this could fit into the active site. And so, again, it doesn't happen all the time, but we have detected xylose modifications on a few EGF repeats in nature. So the second modification is between cysteines 2 and 3. If you see this sequence, a cysteine, 4 amino acids here, which can vary widely, and a serine or a threonine immediately adjacent to the third conserved cysteine, we can predict pretty accurately that there's going to be a fucose in alpha linkage attached to the hydroxyl group of the serine or threonine. That's added by proteinofucosyltransferase 1, which is POFA1. The fucose can be elongated at certain sites with a beta-1,3 glycnac, and these are the fringe enzymes that Ku referred to. I'll be mentioning, why they're called fringes in a little bit. And then because it's a beta-link glycnac, it can be elongated by galactose and sialic acid by enzymes that modify N-glycans and other O-glycans. But the fringe enzymes in POFA1, they're unique to EGF repeats. As far as we know, they do not modify any other protein domains. The third modification is the one that's actually most recently identified. It's between the cysteines 3 and 4 of the EGF repeat. Again, this is a putative consensus sequence, and I'll explain what I mean by that in a little while. But again, there are specific residues that have been found in most of the sites that we've mapped so far. Again, this is a serine-only modification, and it's being added by homologs of poglet1. Remember, poglet1 is modifying a serine over here between cysteines 1 and 2. These are different enzymes, but they're homologs called proteinoglycosyltransferases 2 and 3, and these are partially redundant enzymes. They add, a glucose to the hydroxyl and the serine. Note that it doesn't get elongated with the xylose, as we have not seen xylose modifications at this site. And so the first site here does get elongated, and in fact, most of the glucoses get elongated with xyloses, but here we haven't seen any of that. So that's another difference in that glycan. And then finally, there's in between cysteines 5 and 6, at this, again, putative consensus sequence, at a serine or threonine in this position, it can be modified with an extracellular O-glycnac. So again, this is completely different from the nuclear and cytoplasmic O-glycnac modifications that Ku was referring to, that Jerry Hart's lab identified. It's a different enzyme because it's in the secretory pathway, and so it's called the EGF domain-specific O-glycnac transferase, or EOGT. Again, it does add a beta-link glycnac to the hydroxyl of the serine or threonine. And again, because it's a beta-link glycnac, when it goes through the Golgi apparatus, it can also be modified by galactose-sialic acid. But the EOGT enzyme itself is unique to EGF repeats. So Tetsuya Okajima is going to be giving a talk about the EOGT, the extracellular O-glycnac, on Monday morning at 10 a.m. So if you're interested to learn more about that, I encourage you to go hear Tetsuya. So again, it's amazing that there are actually 4 different O-glycans that have evolved all of these enzymes for these protein modules. A small number of EGF repeats have more than one. Again, we've not identified any individual EGF repeat that has all 4. I think there's 1 or 2 proteins that have 3 of these modifications in an individual EGF repeat. But most EGF repeats will just have 1 or 0 of these O-glycans. So now just into the biological functions of the repeats. So again, the nice thing about having consensus sequences is that you can do database searches and predict what proteins might be modified. And so if you take the consensus sequence for O-glucose modification, the poglet1 site, in an EGF repeat, there are about 40 human proteins that have that consensus sequence. And the ones that are highlighted in red are ones that have been confirmed that they are modified. The initial proteins identified with the O-glucose trisaccharide were factor 7 and factor 9. These are clotting factors in the blood. They've got single EGF repeats. But we really don't know what the biological function is for that. When I started working on these O-link glycans on EGF repeats, we were fascinated that the Notch receptors, which there's 4 Notch receptors in humans, had more O-glucose sites, EGF repeats with O-glucose sites than any other protein in the database. And so we turned our attention to studying the modifications in that context. We can do the same thing for the O-fucose modification site. Again, when you do a database search with that sequence in the context of an EGF repeat, there's about 100 proteins that are modified, that are predicted to be modified. Again, the red ones are the ones that have been confirmed. The first protein that was identified with this was tissue type plasminogen activator, Genentech. It's a drug, a clot-busting enzyme. And they identified it when they were characterizing their drug. There was an extra 146 MASH units on the EGF repeat. And then some clotting factors were identified. But again, all 4 of those Notch receptors also have modifications or predicted sites. And again, there are more O-fucose sites on the Notch receptor than on any other protein in the database. And so again, those were the things that made us start to look at the Notch signaling pathway. And so I'm going to, for those of you that are not familiar with Notch, I'm going to give you a brief primer on what it is. Notch has been studied for literally over 100 years. It was originally described by T.H. Morgan back in the early part of the 20th century. He was mutagenizing flies, and he was looking to see whether or not phenotypes that he observed obeyed a Mendelian inheritance patterns. And so he would oh, did I do that? He identified some flies that had a portion of the wing missing, like this. And so Drosophila geneticists say they name their phenotypes based on what they see. And so this was the loss of the wing tissue. They call it a notch because it looked like a notch in the wing. Interestingly, they only saw that notch on female flies, not male flies. And after doing many crosses, they realized that it was a sex-linked lethal. So the male flies were dead, and the female flies had the notching phenotype. 70 years later, the gene that's mutated in those flies was identified by Spiros Artabanos-Sasankis' lab at Yale. He's currently at Harvard. And shown to encode a very large cell surface receptor. And since then, Notch receptors have been identified in all metazoans, and they're essentially they're essential for development in metazoans. So again, metazoans being multicellular animals. Dysregulation of the pathway results in a variety of diseases, cancers, in particular T-cell acute lymphoblastic leukemia and squamous cell carcinomas. Vascular disorders, there's a nasty one called CADASIL, which is a mutation in Notch3. The most common form of congenital heart defects in humans is caused by mutations in the Notch pathway. And Alagille syndrome is a disease where the development of the biliary trees in the liver is compromised, and for children that don't get a liver transplant by about age 5, they'll probably die. Actually, Hamid Jawar-Nijad is going to be giving a talk about the role of glycosylation of Notch in Alagille syndrome as possibly a therapy. So it's a really interesting pathway, and I'm just going to briefly go through how it works. So the protein begins in the endoplasmic reticulum, and that's where the O-fucose and O-glucose modifications are added in the ER. As you go through the Golgi, that's where the fringe glycosylation occurs, and additional the galactose and sialic acid. And then the protein is at the cell surface, and the unique thing about the signaling pathway is that it's adjacent cell signaling. The ligands themselves are transmembrane proteins, so they have to be on an adjacent plasma membrane. Once the ligand binds to the extracellular domain of Notch, there's an endocytic event happening where the ligand pulls on the Notch, the extracellular domain, that actually causes a conformational change very near the membrane, and it exposes a protease site that is cleaved by a cell surface atom protease, atom 10 or atom 17. The extracellular domain of Notch actually gets transcytosed into what's called the signal sending cell. That's followed very, very soon with a second proteolytic cleavage catalyzed by gamma secretase, which is actually intramembrane proteolysis, but it releases the cytoplasmic domain of the Notch receptor, now a soluble protein. It has nuclear localization signals in it, and so it goes into the nucleus, and it binds the protein's transcription factor called CSL, which is in the already bound to the DNA. In the absence of the Notch intracellular domain, or NICD, this is a transcriptional repressor. But when the NICD comes in, it kicks that off and it recruits other proteins and activates transcription. So it's, again, a really interesting pathway, partly because it's adjacent cell signaling as opposed to many, many other types of receptors that are binding soluble ligands. Also, you destroy the receptor every time you activate it. And so if you want to maintain a signal, you have to kind of continually synthesize the Notch receptor. So there's one notch in Drosophila. There are 4 notches in mammals. The notch 1 and notch 2 are most similar to the Drosophila form. Notch 3 and notch 4 have shorter extracellular domains. There are 2 families of ligands that can activate Notch. There's the serrate-jagged family. Again, Drosophila has serrate, one copy in that family, and the jagged 1 and jagged 2 are the serrate homologs in mammals. In the delta family, there's 1 delta in flies, and then there's delta-like 1, delta-like 3, and delta-like 4, all of which can activate Notch. Just to kind of zoom in and look at the structure of the Notch receptor, here's the transmembrane domain. So this is the intracellular domain and the extracellular domain. This is where those proteases cut, and this is that negative regulatory domain that gets unfolded so that you can expose the site 2 site. Here's the nuclear localization signals for going into the nucleus for the Notch intracellular domain. And this is the transactivation domain. So it's what recruits other proteins to activate transcription. So we're going to be focusing on this region of the receptor. And the prototypical notches have 36 tandem EGF repeats. That's what each one of these little rectangles are. And again, just summarizing a whole bunch of work, we've done a lot of site mapping of the O-fucose and the O-glucose glycans. There are also O-glycnac glycans on here too, but it's hard to put them all in the same figure. So in this figure, the ovals are EGF repeats. If it's colored red, it's got that POFIT1 consensus sequence. If it's colored blue, it's got the POGLIT1 consensus sequence. And when it's striped, it has both. And so we've done glycoproteomic site mapping on both. This is mouse Notch1. We've also done human Notch1. And you see wherever you see a blue oval, it's got a red triangle there, and that means we know that there's a fucose there. Okay, and there's 2 sites, 2 EGF repeats that have the putative consensus sequence, but they are not modified. And so but 17 of the 19 EGF repeats are in fact modified, which again shows that consensus sequence is quite accurate. All of the O-glucose modification sites are in fact modified. So again, that consensus sequence is quite accurate. So again, it demonstrates that the extracellular domain is heavily decorated with these O-link glycans. So again, summarizing several years of work from a number of different labs, we know that the O-glycosylation of the EGF repeats in Notch are essential for its function. These are mainly knockout studies. So if you knock out the POFIT1 in a mouse or OFUT1 in flies, it's just a different abbreviation for the same enzyme. Results in strong Notch-like phenotypes. These were published back in the early 2000s, and so they're phenocopying Notch. And so if you knock out Notch1, you get the same kind of phenotype as you get with a POFIT1 knockout. Same thing for POGLIT1. Again, it's called RUMI in flies, because again, it was identified at a different time. And again, results in strong Notch-like phenotypes. The original publication was on fly RUMI in 2008, and then the mouse knockout was in 2011. So that's the addition of that first sugar, the glucose or the fucose, is essential. Elongation, so adding the xyloses or adding the glycnac being added by the fringe proteins. They will modulate Notch activity. They're not required for Notch activity, but they can either increase or decrease Notch activity. Especially fringe can actually do either. It can increase or decrease, but it depends, as I'll mention in a little bit, which ligand is being expressed in the system that we're looking at. So again, the demonstration that the fringe proteins were glycosyl transferases came out in two Nature articles in the same issue. That's what Henrik and I were competing for that. So Henrik's lab and my lab both demonstrated that that the fringe enzymes, that the fringe proteins are glycosyltransferases, came out at in the same issue of Nature. So addition of xylose to the O-glucose tends to decrease Notch activity. And then again, I'm not going to say anything more about O-glycnac other than the fact that it's known to modulate Notch activity. And again, Tetsuya Okajima is going to be talking about that on Monday morning. So this is just one figure from Pamela Stanley's 2003 mouse knockout of POFIT1. And again, as I mentioned before, this phenotype phenocopies the Notch1 phenotype. So in each of these panels, there's on the left, there's a wild-type mouse, and the right is a POFIT1 knockout. And so we see things, this is a defect in cardiogenesis. You get this bubble around where the heart's supposed to be. Semitogenesis, and we'll talk more about this, but semitogenesis, the formation of the spinal column and the rib cages, and that's a Notch-dependent pathway. Vasculogenesis is just pretty obvious. Neurogenesis, these are tissues that all require Notch activity at some point in their development. And when you knock out POFIT1, you see the same phenotype. So again, strongly indicating that Notch requires the addition of O-fucose to be functional. And again, a decade of in vitro studies demonstrated the same, that if you don't have an O-fucose on the EGF repeat of Notch, then it doesn't bind its ligands and won't signal. It won't activate. Again, this is the paper that Tetsuya Okajima's lab published that O-glycnac it can modulate ligand binding, and it also affects vascular development in mammals, especially in retinal angiogenesis assays. Okay, so why is fringe called fringe and not beta-3? So I'm a biochemist. I purify the O-glycnac transferase, and so I name things, what they do catalytically. If I had discovered the fringes before anybody else had, I would have called them beta-3 glycnac transferases A, B, and C. But somebody before had identified the gene. And so back in 1994, Ken Irvine, when he was a postdoc in Eric Wischhaus's lab, was looking for modulators of the Notch pathway. So he was doing the same thing that T.H. Morgan was doing. He was mutagenizing flies and then looking for a Notch phenotype. And then he'd do complementation analysis to see, okay, I don't want to be mutating the Notch gene, but I see if it's a separate gene. And so he found several different genes. And one of them he called fringe because the fringe of the wing is lost. And again, Drosophila geneticists like to name things based on what they see as a phenotype. So that was in 1993. In 1997, 3 mammalian homologs of that gene were identified in mammals. And again, these were geneticists doing this. Again, this is before we demonstrated, because the demonstration that they were actually enzymes was in 2000. So they gave them these great names, lunatic fringe, manic fringe, and radical fringe, which again, I really like. And just to show that these are also biologically important, not just that the addition of the fucose, but the addition of the glycnac to the fucose actually has an important biological role, is that if you knock out lunatic fringe in mice, you get a severe defect in semitogenesis. So remember, when you knock out the fucosyltransferase, you get a dead mouse. So this mouse actually, I forgot to mention that these mice die right around embryonic day 10. So for those of you who don't work with mice, it takes 21 days for gestation of a mouse. And so this is halfway through gestation. So it's very early in embryogenesis, and you're having a really severe and catastrophic event happening. These mice are alive. This is a lunatic fringe knockout mice. You see it has a really short, stubby tail, and this is a littermate that is heterozygous. And so you can see there's something wrong with its spinal column. If you take those mice and you dissolve away the tissues, this is what a normal skeleton should look like in a mouse with a fully formed rib cage. You can see that the spinal column is totally screwed up, and especially the ribs About 50% of these mice die at birth because the rib cage is not strong enough to hold the diaphragm, and so they really can't breathe or can't breathe well. And so it's a very severe defect. But again, remember, when you knock out POFA1, you got cardiogenesis and defects in cardiogenesis and vasculogenesis, and those things don't happen. So in a lunatic fringe knockout, which means that the fringes are affecting notch activity in certain contexts, but not others, whereas the O-fucose modifications and the O-glucose modifications are affecting notch everywhere and in all contexts. There's also human diseases, a congenital disorder of glycosylation, inactivating mutations in lunatic fringe, causes severe vertebral segmentation anomalies in patients. And the disease is called spondylocostal dysostosis type 3. Again, there are several different types of this, but they're all genes involved in the notch signaling pathway that cause this mitogenesis defect. This is an MRI of the chest cavity of a patient with this. And again, these are supposed, the vertebrae should be stacked right on top of each other, and you can see they're kind of cattywampus there, and so it results in a very severe scoliosis in the patients. Again, just a quick summary of a decade and a half of work to what is fringe actually doing. It's going to allow notch to discriminate between the two ligand families. So again, there's the delta family and the jagged family in mammals, and in a cell where there's no fringe expressed, there's just going to be that O-fucose monosaccharide on it. And both ligands will activate that receptor about equivalently. But if there's a fringe expressed, specifically, in particular, lunatic fringe, you'll get some of those O-fucose residues modified with a GLICNAC. And what that does is it enhances the signaling, the activation from the delta ligands, but it reduces the signaling from jagged. It's really interesting. The same modification is having opposite effects on the ligands, and we're still. I mean, we know how the modification affects the increase in delta signaling, but we're still working on the molecular mechanism for how it inhibits signaling from the jagged receptor. It's also just a really good example of when a receptor is expressed in a different cell type that has different glycosyltransferases that modify their extracellular domains, it can modify the signaling process. And so it's a really good example of that concept. There are several congenital disorders of glycosylation which result from mutations in these enzymes that are modifying the EGF repeats. For POGLIT1, there's two. There's a Dowling-Dagos disease, which is a skin disorder. This is an autosomal dominant disorder, which means they only need one mutation in one allele of POGLIT1. There's also more recently a limb-girdle muscular dystrophy. This is a progressive disease in the muscles between the hips and the shoulders. This is automotor recessive, so you need to have two inactivating mutations, and it's not completely inactivating. There's about 5 to 10% of enzymatic activity left in those patients. But again, it causes a muscular dystrophy. POFA1 mutations also cause a different type of Dowling-Dagos disease, the same skin disease as the POGLIT1 mutations, again, also autosomal dominant. EOGT causes a really nasty disease called Adams-Oliver syndrome. I would have shown you pictures, but they're really quite grotesque. And it's autosomal recessive. And then, as we just talked about, mutations, inactivating mutations in lunatic fringe cause spondylocostal dysostosis type 3. So again, these are all rare diseases, as Henrik mentioned, that single gene loss of function of individual glycosyltransferases. The more common diseases are cancers. I mean, many of these enzymes are either overexpressed or underexpressed in cancers because Notch is involved in cancer. And if actually Notch can play either a role as an oncogene or a tumor suppressor, depending on the cancer. And so modifications that will either increase or decrease its activity, which is what these glycosyltransferases do, are implicated in a wide variety of cancers. So I haven't talked about this novel modification. Remember, there's two O-glucose sites on an EGF repeat. So we talked about POGLIT1, and that's heavily involved in notch signaling and notch function. But this modification we just identified a few years ago. So the genes were, the glycosyltransferases were identified in 2018. And again, it's interesting that there's two of them, so they are partially redundant, both biochemically and biologically, as you'll see. And they add this beta-linked glucose to a serine in this position. They don't modify the other site at all. And again, POGLIT1 will not modify this site. And again, it doesn't get elongated with xyloses. So again, we do have a putative consensus sequence. We're still working on this, and you'll see why in just a minute. But the first several sites that we mapped, all of these amino acids between cysteines 3 and 4 were there. And again, the serine is in this position. So doing a database search, there's about 50 proteins that have that consensus sequence in the context of an EGF repeat. And again, the things we looked at first were the notches. Again, they all have at least one EGF repeat with that consensus sequence. But again, it's only one or two. But then when we looked at some of these other proteins, especially the fibrillins, fibrillin 1 and fibrillin 2, they had like 20 EGF repeats that had that consensus sequence. And so we decided, well, maybe we should look at the fibrillins to see whether they really are modified that much. And so just to remind you, if you don't know, the fibrillins and these latent transforming growth factor beta binding proteins, or LTPPs, are extracellular matrix components. So fibrillins are the major structural component of elastic microfibrils in the matrix. They play important roles in TGF-beta signaling, and mutations in fibrillin 1 cause Marfan syndrome, which is actually a fairly common for rare diseases. It's one of the more common rare diseases. And it has specific defects, bone growth defects. I mean, people with Marfan syndrome are really tall, actually. Many people think that Abraham Lincoln had Marfan syndrome. It also causes it has aortic aneurysms, and so most of the patients die with aortic aneurysm. So again, it's an important, they're important proteins for elastic tissues, in particular, like the aorta, the heart, the lung. And then the LTPPs, they are actually, they interact with the fibrillins in the microfibrils, and they regulate the bioavailability of TGF-betas. So the fibrillins are just huge proteins, and they've got 47 EGF repeats, both fibrillin 1 and fibrillin 2. LTPPs, there's 4 different LTPPs, and they have between 15 and 20 EGF repeats in each of them. So a couple years, 2 or 3 years ago, we decided to analyze just the glycoproteomics site mapping of fibrillin 1, fibrillin 2, and LTPP1 from humans. And again, the ovals are the EGF repeats. If it's colored blue, it has that putative consensus sequence in it. And again, when you see a blue circle above it, it means we know that there's a glucose attached to that. And again, the stoichiometry, if it's full blue, it means it's completely stoichiometric, and if it's hashed or something, it's a little bit less than that. So the interesting thing is that we saw more sites modified than we knew with that consensus sequence. I mean, there's this EGF14 here, it's modified 100%, but it doesn't have all of those amino acids from the consensus sequence. And so we're working right now on which of these amino acids is actually essential. I mean, they're conserved in many EGF repeats, but we don't know whether they're essential for glycosylation, and so that's a work still in progress. So again, I'm not going to go into much of the detail of the biological function of them because we're still working on that, but we have generated knockout mice, and single knockouts of either POGLIT1 or POGLIT2 or POGLIT3 are viable and fertile, which again is, I mean, I mentioned before, biochemically they're partially redundant, and so they both can modify most of the EGF repeats. They have preferences, but nonetheless, this kind of demonstrates that biologically they're redundant. But if you knock out both of them together, and this is again unpublished data, we actually just submitted this. They're neonatal lethal, and that means that they're dying shortly after birth. And again, if you knock out the fibrillins or the LTPPs, you get similar phenotypes. And so again, we think that the fibrillins and LTPPs are the major biological target for these, at least in early development. So And again, the double knockouts, as when you knock out fibrillins, they're small, they have lung defects, and that's what we think that they're probably having trouble breathing. And then syndactyly, which means that their digits and their paws don't separate. So again, those are characteristics of fibrillin knockouts. Okay, so that's the EGF repeats, and I'm going to move on to the thrombospondent type 1 repeats. Hopefully I've got a little time. Again, so we'll start with the structure and biosynthesis of the TSRO glycans, and also just what is a TSR. These are a little bit bigger than EGF repeats. They still have the 6 cysteines. So these are 40 to 70 amino acids in length, and you can see they're just kind of they're elongated, sort of like an EGF repeat. But the disulfide bonding pattern is different. There's group 1 and group 2 TSRs. The three-dimensional structure is pretty much the same, but the positions of the cysteines are different. So in group 1, it's 1, 5, 2, 6, 3, 4 is the disulfide bonding pattern, and group 2 is 1, 4, 2, 5, 3, 6. But again, as I said, crystal structures show that the three-dimensional structure of a group 1 and a group 2, you can almost overlay the backbones. Again, the other amino acids can be quite variable, and they're found in dozens of cell surface and secreted proteins and metazones. There are many, EGF repeats, they're literally in hundreds of proteins in mammals, but there's less than 100 proteins that have TSRs. And again, they, just like EGF repeats, they often occur as non-identical repeating domains. So TSRs can be modified by C-mannose and O-fucose. Again, the consensus sequence for C-mannose is WXXW or WXXC. I'm not going to say anything about this, but Hans Wacker is going to give a talk Monday morning at 9:30. And so if you want to hear more about C-mannosylation, go to his talk, okay? So I'm just going to talk about the O-fucose modification that's added by protein O-fucosyltransferase 2 to this serine or threonine. It's between the cysteines 1 and 2 or cysteines 2 and 3, depending on whether you're a group 1 or group 2 TSR. You need 2 amino acids here and a serine or threonine there. We can again predict pretty accurately that if you have that sequence, it's going to be modified with an O-fucose. POFA2 is a homolog. It's 30% sequence identity with POFA1. So it was predicted to be modifying something like EGF repeats. And then it can be capped with, excuse me, a beta-1,3 linked glucose. Remember, the fringe enzymes add a glycnac beta-1,3 to a fucose, and this is a glucose being added to the same position on the fucose. The beta-3 glucosyltransferase are homologs of the fringes. It's interesting that these are completely separate pathways, so POFA1 will not modify this serine or threonine, and fringes will not modify a fucose in this position. And so they're specific for EGF repeats, whereas POFA2 will not modify an EGF repeat, and the fringes will not, the beta-3 glucosyltransferase will not modify an O-fucose on an EGF repeat. And so again, these are completely separate pathways. There's no crosstalk at all. So biology of TSR glycans. So again, we've got a consensus sequence, and so we can do database searches. We're searching, this sequence, cysteine, XX, serine or threonine, cysteine, in the context of a thrombospondin repeat or a TSR. And there's 49 proteins in the human genome that have that. The ones colored in red are ones that my lab or others have demonstrated are in fact modified at those sites. You can see that there's a large series of these ADAM TS proteins. Probably more than half, about half of the proteins that are predicted to be modified are in this family. These are extracellular proteases. They remodel extracellular matrix. They all have a protease domain, and they've got a variable number of the thrombospondin type 1 or the TSRs in purple ovals. So as few as 1, others up to 15 TSRs. So again, we wanted to know what the biological function is, and sort of the first thing you want to do is you knock it out in an organism, and we knocked it out. This is work done in collaboration with Bernadette Holtner, who's collaborated with me on essentially all of our mouse projects. So we knocked it out, and the mice died actually quite early at embryonic day 7.5. And so this is even earlier than the Notch phenotype. So again, it just also demonstrated that it's not affecting the Notch pathway. So this is a wild or heterozygous littermate to a homozygous knockout, and you can see it just. I mean, this is the embryo, and it just doesn't look. It looks funny, and it actually doesn't proceed after this. And at embryonic day 7.5, it's a really important moment in development. It's when gastrulation occurs, and gastrulation is when the 3 germ layers are formed: endoderm, ectoderm, and mesoderm. If you don't form the 3 germ layers, you're not going to form any tissues. And so these embryos get resorbed in the uterus a day or so after this happens. So we were interested in which of those target proteins would be is the cause of this, because obviously there's going to be one of the proteins that's modified that will give you that phenotype. And again, we've been collaborating with Sunil Apte at Cleveland Clinic, who is sort of the world's expert on the ADAMTS proteases, and he had just knocked out a whole bunch of them, but only one of them gave an embryonic lethal phenotype. And so he sent us the mice, and Bernadette's lab looked at them in detail and showed basically that the same phenotypes occur, that the ADAMTS9 mice die at the same time during gastrulation. And so we get the same phenotype as the POFA2. So it strongly suggests that when you don't add the sugars to the ADAMTS9 protein, it's non-functional. It doesn't tell us why, but it tells us that's a really important substrate for POFA2. There's also a congenital disorder of glycosylation related to this modification, and it has to do with the glucosyltransferase. So it's called Peters plus syndrome. So patients with Peters plus syndrome have four recurring phenotypes. One is an eye defect called Peters anomaly, where in development the cornea and the lens don't separate. And so it causes a cloudy eye, and this can be surgically repaired, but if it's not surgically repaired, then they're going to be blind for the rest of their lives. And then they have bone growth defects. So there's a craniofacial defect, long bone defect, which means they're going to be short and have short arms. And then they've got short fingers called brachydactyly. And so those were clinicians could identify patients with Peters plus based on those 4 phenotypes. But in addition, they often occur with cleft palate, cardiac and urogenital system disorders, hydrocephalus, and developmental delay. The gene has got 15 exons, and there are mutations scattered across the 15 exons that can cause the disease. And in particular, there's several in exons 12 and 13, which are actually in the catalytic site. This D349 is actually the catalytic aspartate that is required for catalysis. So again, we also made a knockout of the beta-3 glucosyltransferase in mice, and it recapitulates most of the phenotypes except for the eye defects. I mean, they have an eye defect. Actually, the eyes don't form very well. Half of the mice, the eyes are not completely there, but they don't have that Peters anomaly. So we think that probably it's a little bit more severe of a phenotype in a mouse than it is in humans. But the bone growth defects are there. This is a wild heterozygous littermate of a knockout of beta-3 glucosyltransferase. You can see they're smaller and their weights are smaller. You can see that there. You can see there's a rounded kind of dome head, and that's indicative of hydrocephalus, which again most of the mice have. The snout is shorter, so there's a craniofacial defect, and so these are all similar to the defects that we see in the human disease, and we're using this mouse as a model now to look at this disease in more detail. So for the last few minutes, I just want to talk about common mechanisms by which glycosylation of these EGF repeats and TSRs affect the function of the proteins that they have. This is not meant to be that these are the only mechanisms, but these are the ones that we've observed so far. And one of them is extracellular, which is altering binding effects, and we've already talked about that with the fringes. But I'll show you a few other things that show that they function extracellularly. But then we'll also talk a little bit more time on intracellular functions. We think they're involved in the quality control of folding of the EGF repeats and the TSRs. So again, as I mentioned before, fringe modifications of the extracellular domain of Notch can alter the activation of the receptor. It increases activation by delta ligands, and it decreases activation by jagged. And again, we don't really know the mechanism for jagged, but the mechanism for delta is increased binding. I mean, when you add these sugars to Notch, the affinity for delta-like ligands goes up. And so it's a perfect explanation for why the signaling goes up. So that's one example of extracellular function. Also, Chris Garcia's group at Stanford, who's a crystallographer, has made co-crystal structures between a portion of the Notch receptor, Notch's, the EGFs 11, 12, and 13, which are part of what's called the ligand binding domain of Notch, and two ligands, delta-like 4, there's, again, the portion that binds to Notch, and also a second one where there's a bigger portion of Notch 1, EGFs 8, 9, 10, 11, and 12, and a portion of the jagged receptor. And when you look in detail at the interfaces between the receptor, the Notch receptor, which is in purple, I guess, and in either the blue or the green, on EGF12, there's a fucose, an O-fucose, and the O-fucose is actually, for both the jagged and the delta complexes, the fucose is actually physically touching the ligand. And again, when you mutate that residue that has that O-fucose on it, it significantly reduces the binding affinity between Notch and the ligand. And in fact, Ahmed Jafarnejad's group has mutated that residue in the endogenous locus of Notch in flies, and Pamela Stanley's group has mutated that amino acid in the Notch1 locus of endogenous locus in mice, and it causes embryonic lethality. So just the single mutation, eliminating that one fucose residue, is enough to actually abrogate Notch function in vivo, which is really quite spectacular, I think. Again, very recently, the Garcia group, which is, again, at Stanford, has done a co-crystal structure of the TSR3 of brain-specific angiogenesis inhibitor, BAI1, and shown that the fucose is in contact with a reticulon 4 receptor. So these are receptors that are in the nervous system. They are expressed on neurons and glia cells. The binding regulates dendritic arborization, axonal elongation, and formation of synapses. And so it's a very important function in the nervous system. And they're looking at this TSR3 on the BAI protein, which has an O-fucose on it. And so it's pink here. There's the threonine, and it gets modified. There's the fucose, and there's the glucose, and this portion of the receptor. And again, you can see that there's multiple contacts between the receptor and that O-fucose. And again, if you mutate that threonine, it removes the fucose, and binding disappears. And so the binding between the receptor and the BAI1 is gone. It's another example of, and I mean, we think this is going to be going on for many of the proteins that are modified with O-fucose and O-glucose. It's just hard to find them. So you find them sort of serendipitously like this. I mean, they had no idea that the fucose was going to be important in that binding event until they did the co-crystal structure. And when they did that, then they actually sent it to us, and we made a mutant and showed that the binding disappears. And so it's really, I think we'll have a lot more of these examples that'll come up in the future. So now I want to talk a little bit more about the intercellular functions, because it's a little bit more complicated to explain than just a simple binding event. All of these enzymes, the ones that add the sugar directly to an EGF repeat or a TSR, so POFOT1 and 2 and POGLITs 1, 2, and 3, and even EOGT, which I don't have data for here, but it does the same thing. They only modify folded domains. Okay, we learned this first with POFOT2 back in 2006, because we had to design an assay to prove that that the gene encoding POFOT2 actually was modifying a TSR. We made a TSR in bacteria. Bacteria don't have the enzymes, and so they're going to be naked, and we could show by biophysical methods that it was folded. It had the right disulfide bonding pattern, and so we could use that as a acceptor substrate in an in vitro assay. But if we took the TSR and just unfolded it, we just broke the disulfide bonds with DTT and modified them with iodoacetamides, that wouldn't reform. It's not. And again, if you take a peptide that has the consensus sequence, it's not a substrate in vitro. And so this was the first indication that these are actually recognizing something that's folded and not folded. We see the same thing for POGLIT1 and POGLIT2. They're modifying EGF repeats. If you reduce and alkylate the EGF repeats, they don't modify it. There have been several co-crystal structures shown of these enzymes. So Jim Rennie's lab has done a co-crystal of POFOT1 and POGLIT1 co-crystallized with an EGF repeat. And then Ramon Hurtado Guerrera's group in Spain has done a co-crystal structure of POFOT2 and a TSR. And this actually, the crystal structures help to explain why you need both a folded domain and a consensus sequence, because you have to localize the hydroxyl group that is going to be modified precisely in the active site of the enzyme, and which has to be within angstroms of the nucleotide sugar, where this is going to perform a nucleophilic attack on the sugar that's going to be transferred. And so the other thing you can see is that there's a cleft in each one of these enzymes that's big enough for a folded domain to fit. So this is just emphasizing that the enzymes recognize a folded domain with the proper consensus sequence that it can modify. We were wondering that it seems like these enzymes are they're all soluble enzymes that are located in the ER, which is the folding compartment in the secretory pathway. They also can distinguish folded from unfolded EGF repeats and TSRs. So just a couple minutes. Serving, and so we think they're serving as folding sensors. So the question is whether or not, because if you screw up something in the quality control pathways in the ER, it's going to affect the ability of those proteins to get out of the ER and get to the cell surface or be secreted. So the question is that happening? And so I just go through something real quick. So we've made knockout POGLIT1 and or POFOT1 knockout cells in HEX. The HEC293C cells, they express endogenous Notch1. And so this is a flow cytometry reading where the red line shows the Notch1 antibody and the black line is a control antibody. And so you can see on a wild type cell there's Notch1 on the surface. If you knock out POFOT1 or POGLIT1, you can see a more than 50% reduction in cell surface Notch1. If you knock out both of them, you essentially lose Notch on the cell surface. So this is really strong data suggesting that they are involved in quality control. The other, Kenjiro Matsumoto, who was a former postdoc in my lab, has actually got a talk about the trafficking of Notch in POFOT1 knockout cells Friday at 10:20. So if you want to hear more about that, you should go. We see a similar thing with the ADAM-TS proteins, that if you knock out POFOT2 or the beta-3 glucosyltransferase, you see reductions in secretion. And in the interest of time, we'll not go through that data. We also think the mechanism is that when you add these sugars to the domains, they stabilize the folded domain. the sugars, if you look at them in NMR or in crystal structures, the sugars are laying down on the face of the EGF repeats or TSRs with multiple contacts around it. And if you mutate the amino acids that they're interacting with, the stability is reduced. And so we think they're adding stability to the domains in the ER. It's sort of a stamp of approval. You know, you get the disulfide bonding pattern right, then you put a sugar on it, and it stabilizes, so it's not going to be, the disulfide bonds are not going to be shuffled anymore. So again, mechanisms by which O-glycans on EGF repeats or TSRs affect protein function, just mediating protein-protein interactions, and also quality control and trafficking. And the different proteins that are affected, mainly for the EGF repeats, it's Notch, but there are others that I haven't gone into. And for POCA2 and 3, it's the fibrillins, and for POFIT2, it's mainly those ADAM-TS proteases. So these are the folks that have done the work. Stony Brook, which is where I used to be, is where Bernadette Holdner is, and we've had a long-term collaboration on the mouse knockouts. Hideyuki Takeuchi was a research associate in my lab for 10 years, and he's just recently moved to Shizuoka for his own lab. These are the nice people that give us the money. This is a recent retreat with Bernadette's lab at the CCRC, and I'd be happy to take questions.
Thank you, Bob. It's a fascinating journey and a story. And although it's getting late, but we do reserve 20 minutes for
Yeah.
the question and discussion.
Mhm.
Okay. So anybody, any questions?
And thanks for staying.
Eric?
Very nice, Bob. Is it on? Yes. Can I always wondered about French.
Yeah.
It's true we met, in 2000. It's secreted. And lunatic French is also secreted in mammalian cells. So what's that all about?
Right, so both Drosophila fringe and lunatic fringe have like pre-pro. They have a furin cleavage site that is cleaved when the proteins get into the Golgi, and so they are secreted. They are retained in the Golgi partially, but not for a long time. Manic and radical fringe don't have that furin cleavage site. They're Golgi resident enzymes, so they don't get secreted. It's interesting, I mentioned that mutations in lunatic fringe cause semitogenesis defects, and what it's doing in semitogenesis is that the lunatic fringe levels go up and down and up and down, and they're turning Notch function on and off. Because during semitogenesis, the pre-Semitic mesoderm is pinching off what are called somites, and the somites are going to turn into the vertebrae and rib cages. And so every time you get a modulation of Notch activity going from on to off, you'll pinch off a somite. If you mutate the furin convertase site in lunatic fringe, totally screws things up. The reason is that it has to be a short signal because the message for lunatic fringe goes up and then it goes down rapidly. And in order for it to go down rapidly, you have to get rid of the protein quickly. And so if you don't get rid of the protein quickly, because it's going to get secreted, then it stays too long, and you don't get that pinching. You don't get the semitogenesis happening. So that's one reason for why it gets proteolyzed and secreted. And the same thing happens in Drosophila in wing development, where you need a short burst of lunatic fringe or of fringe activity, and then you need it to be removed. And so it's a good way to. It's like degrading a protein rapidly. Does that make sense?
Oh, absolutely. I'm just thinking there must be a co-regulatory mechanism, because either it's a very bad furin site, or there must be something that at certain times can leave a little more or less, so basically regulate the level of fringe.
Well, I think the main biological event, at least during development, that fringe is involved in is is that turning of the notch on and off.
That's true.
Yeah, so, I mean, I think that's, it's important for it to be highly active, you know, the furin convertase site, so.
So I have a question about the PO group. Okay.
Which one?
PO group.
Two and three?
POGLUT.
Yeah.
Glucosyl transferase.
Yes.
2 or 3.
Right.
It also transfer the xylose.
Yes.
And so how to regulate the one side the xylose transfer and other type of glucosyl
Yeah, I
transfer.
mean, we don't
So-
know yet.
Okay.
So even in the co-crystal structures between the EGF repeats and the POGLET1 enzyme,
Yeah.
they speculated about, you know, what xylose, why it's transferred
Yeah,
to
yeah, yeah.
one EGF repeat or not. Again, I mean, we have not seen it on many EGF repeats when we isolate, when we do site mapping on notches from different tissues. But EGF 16 on notch 2 has maybe, you know, 20 or 30% O xylose and the rest O glucose. So it's a mixture.
So
No,
it
though,
may be the concentration of the UDP xylose.
No, I think it's something in the surface of the EGF repeat that is causing it to be preferentially modified. Because again,
Okay.
there are other EGF repeats that in that same protein, in Notch 2, that are not modified by a xylose at all.
Okay.
The one thing that has also been, we don't know about is it's a beta-linked xylose. And beta-linked xylose is a primer for glycosaminoglycan biosynthesis. And so we have been looking for evidence that that actually gets elongated. Again, when we see the O xylose, it typically gets modified with
In this case, every time, so that just stop in the xylose, no
Yes.
elongation.
Well, no, it does. I mean, so it gets xyloses put on top of it. And so
Okay.
the glucoside xylosyl will actually transfer a xylose to this, so you get a triple xylose modification. So we
Okay.
think it's probably blocking the GAG elongation.
Okay.
But again, the other glucose, the newer one, POGLET 2,3, it also, again, in vitro, it can it can actually transfer a xylose. We don't know whether it's that happens in vivo. We haven't seen any sites based on glycoproteomic site mapping that have just a xylose there. So I'm not sure that actually happens in vivo.
Thank you.
Hi, Bob, you show us why it is called fringe and notch. So why is it called Rumi?
Rumi. So this is so Hamid Jafarnijad named this. When they had identified, again using just mutagenesis of flies, another gene that modified gave notching phenotypes in wings, and so this was in flies. And Hamid is Persian. He's Iranian, and he likes there's a poet called Rumi, and he likes the poetry. Also, he says there are some escapers of the mutation that survive to adulthood, but they do have a weird behavioral. They spin around with their wings up in the air, reminding him of whirling dervishes. Whirling dervishes, when they're spinning around, they recite Rumi poetry. And so that's why he called it Rumi. Again, this was a collaboration between my lab and Hugo Bellin's lab at Baylor. And they knew it had to be a glycosyltransferase because it had this CAP10 domain, which is a glycosyltransferase domain in Cryptococcus neoformans. But they were thinking it was going to be modifying N glycans or something. But we had been looking for the glucosyltransferase we were trying to purify the glucosyltransferase from tissues, like bucket biochemistry again, because we were having no success. But I went to a Gordon conference and talked about this, and Mike Tiemeyer was familiar. He had gone to the Drosophila conference just before that, and he said, "Oh, you need to talk to Hugo Bellin because they have some kind of glycosyltransferase that's regulating notch activity, and they don't know what it is." And so I called Hugo. They sent me the construct that week. The next week, we showed it was the glucosyltransferase because we had the assays and everything. So that's why you go to meetings. You hear somebody saying, "Oh, we think we have your gene." So. So
Yeah.
that's the story for Rumi.
So Rumi has a more poetic name.
Yes, yeah, exactly. Right, right.
Alice?
You showed us deficiency of these enzymes can cause serious medical problems. So, can you give us some perspectives about the development of therapeutic approach, like enzyme replacement therapy or messenger RNA therapy?
Yeah, I mean, the deficiencies in the enzymes, like I said, are pretty rare diseases. Unfortunately, they are needed for early stages in development. So like gastrulation or semitogenesis, things like that. So I mean, you would have to have identified the defect before you could actually design a treatment for it. So, I mean, it would be great if you could do that. I mean, for cancers, though, because there are a number of cancers like that overexpress POFIT1. What happens when you overexpress POFIT1, again, partly because of this quality control pathway, is that it actually allows more notch to get to the cell surface. And so you get increased notch on the cell surface, which ends up with increased notch activation. And in certain cells, it depends on the cell, that will cause a mitogenic phenotype. It'll cause them to divide when they're not supposed to divide. So inhibitors of the enzymes would be useful, and we've been working on inhibitors for the enzymes for a number of years. And you know, we have some that work, but they have side effects so far. So we haven't cured cancer yet. So.
That sort of leads into the question I was going to ask. Metabolic inhibitors for fucosyl transferases or glucanac transferases, do they inhibit either of those enzymes?
Right, so like
2-fluorofucose
does not inhibit these enzymes at all. Yeah, so I mean, we've demonstrated that because we went through, you know, all the fucose analogs that are available, and none of them inhibit 6-alkynyl-fucose or 6-alkynyl-fucose. They're substrates for the enzymes, and so they will actually transfer that modified fucose to the Notch receptor, and interestingly, that inhibits Notch activity because a modification on the 6' hydroxyl of the fucose gets in a steric hindrance when you look at that co-crystal structure I showed you. And so it will inhibit, but it only inhibits binding to delta, not to jagged. So it's actually it's the only small molecule that has been identified that will inhibit one ligand and not another. The problem is you're actually modifying the receptor, and it's going to be immunogenic to anybody if you treated them for any length of time. So that's probably not going to make it very far. But we use it in the lab a lot because it's a great Notch inhibitor.
Bob, you show a few crystal structures. So far, they are only single domain structures. Anybody try multiple domain?
Well, so those had 2 or 3. So the one with jagged had I think 5 EGF repeats. The one with delta like 4 had 3. It is extremely difficult to make large amounts of protein. I mean, this is why you probably need specialized quality control machinery in the cell to make these proteins. Because if you think of Notch, it's got 36 EGF repeats. Each have 6 cysteines. That's 216 cysteines that have to find their partner before you're going to leave the ER. It's not going to leave the ER until they've found the right partner, because you can't have unpaired cysteines. It screws things up. And so, you know, we think that these enzymes evolved. I mean, I showed earlier that, you know, the enzymes evolved at the same time as EGF repeats and TSRs. I mean, there are no EGF and TSRs in plants or fungi or bacteria. They're only in metazoans, and the enzymes are only there too. So they seem to have arisen at the same time for a need. And I think it's for these proteins that have multiple EGF repeats and TSRs. They just help them to, you know, once they're folded, they put a sugar on them, and that tells the cell, you know, maybe the oxidoreductases are just not going to bind it anymore. So I think it is hard to make large amount of protein. I mean, you can make 2 or 3 domains in bacteria, but you have to, unfold them and then refold them in vitro to get it to work. But it is, to make, all 36 EGF repeats of Notch, nobody's been able to do. I mean, we can do it in small amounts, but not enough to do a crystal structure. I mean, cryo-EM is being...
Does it help?
Well, we're working on that.
Okay.
It's been a long road.
Okay.
Hi, thank you for the informative talk. In the earlier of your presentations, you showed that there are many EGF repeats with the different glycan structures and different sites. So we are curious about how do you determine the structures and the sites for those many EGF repeats in
Right,
the...
that's a good question. So, I mean, the structures were, the first EGF repeat with an O-glucose on it was discovered in 1988 on a I think it was factor 7. And they did structural analysis on the glucose xylose and showed that there was a trisaccharide on that. And then I think the first time that an O-fucose was initially identified in 1991 or 1992 by Genentech as a monosaccharide fucose on an EGF repeat of tissue type plasminogen activator. And then shortly after that, they identified it on factor 9, and it had the tetrasaccharide. So the structures that are common for O-fucose and O-glucose were known by the early 1990s. So that's sort of, I mean, this was before what's known as glycomics analysis. I mean, there was no mass spectrometry going on. It was either you made a whole bunch of it and did NMR, or what we did is we would use radioactive sugars. We would label cells with the radioactive fucose, and then we would beta eliminate the sugars off of the protein, and then run columns to see, how big the sugars were. So learning of the structures of the sugars was mainly done back in the 1990s. To find where they are on the protein, you have to do glycoproteomics. And so we make the protein, or we immunoprecipitate the protein out of a tissue or a cell line, and then we digest it with proteases and run it through a mass spectrometer and look for. I mean, the nice thing about our glycans is that they're well separated. So usually you only get 1 or 2 glycans on a peptide because they're, the EGF repeats or TSRs are 40 or 50 amino acids long. Mm-hmm. And so you're not going to get 2 O-fucoses in a peptide. If you do, it's too big for the mass spectrometer to analyze. So by identifying the peptide, and again, the nice thing is we have consensus sequences, and so we don't even really need to know exactly which amino acid is, because if there's no consensus sequence there's not going to be a modification. So we do glycoproteomics to find out where they are. And again, when you do the glycoproteomics, you see not only the peptide that's modified, but you also see whether it's a mono, di, or trisaccharide. And again, we know the biosynthetic pathway, so we can figure it out from that.
Another follow-up question is, you know what sugars is on each side, but how could you tell the linkages or the alpha betas of those sugars?
Right, so that was what was determined in the 1990s. They both by exoglycosylase digestions and also by NMR, they determined the linkages. So.
Okay, thank you.
Sure.
Okay, so one last question. So you mentioned that the TPA and that have these fucose and that they were the one that discovered first.
Yes.
So does it matter to have that?
Oh yeah. So again, the division at Genentech that developed the clot-busting enzyme, the TPA. They was because it was one of their first multi-billion-dollar drugs. But before the FDA would approve it, they had to give a chemical, how many carbons, nitrogens, oxygens, are in a drug. And so they did massive amounts of mass spectrometry, and they found, this extra 146 mass unit on the EGF repeat, which they were like, wow, that's weird. And they went through, they mutated the site, they did every assay they could do that they knew of, stability, enzymatic activity, retention in the serum, all that stuff, and they saw nothing. It didn't do anything. So nobody knows what it's doing. Again, many of those proteins,
Including
they
the
just-
factor 9, factor 7.
Sorry?
Also the blood coagulation factor?
There have been some studies that O-glucose on factor 7, I think, has a mild effect on its activity, but not like Notch. So,
How about their biosynthesis? Is it affected
biosynthesis
of those
oh,
proteins?
whether or not they get secreted and
Oh,
stuff.
no.
No, they're not. There's not an effect. So
No
again,
effect at all.
usually, we've had some examples where, a lack of glycosylation of a single EGF repeat has a profound effect on secretion. But usually, it's more, multiple repeats that have a bigger effect.
Okay, I think we're here.
That's fine.
And we'll thank Bob again for the fascinating talk.
Thank you.
Thank you. And hope we all learn a lot about protein glycosylation. So we'll continue tomorrow morning. Okay? See you.